Thank you very much, Harvey, for the kind introduction. I would like to begin with a longer list of thanks, first and foremost to Shaul Massry, who, of course, through his generosity, endowed this award. Then to all the members of the selection committee for their impeccable scientific taste in choosing, in choosing this particular topic and this particular trio of laureates. And finally, to the communities at USC and UCLA for the warm hospitality that we have experienced over these 3 days. Now, I did not come to experience this warm hospitality all on my own. I brought with me my doppelganger. This is Dr. Gero. We have many things in common in addition to just the first name. He's also a scientist, a mad one, in this Japanese comic called Dragon Balls. He strives for world domination, just like I do. And if you look carefully at his skull, you can see that it has been replaced with a transparent plexiglass dome, so that the workings of his brain are open to optical control. And this is, of course, exactly what this is about. We modify particular classes of neurons in the intact nervous system genetically so that they can become responsive to light. And these two essential ingredients, genes and photons, have given the field its name. Now, what motivated the invention of optogenetic control was the idea that a technology like this would open 3 experimental doors in neuroscience that had previously been locked. The first is the ability to identify the neuronal causes of behavior. If one could play back activity patterns into the nervous system and in this way recreate perception, action, emotion, thought, then one could credibly claim that one has truly understood the physiological underpinnings of these aspects of our mental lives. It's sort of a biochemical reconstitution approach. This is the philosophy that I took from Jim Rothman. In order to understand, you have to be able to control. And the control you exert, if you're a biochemist, by purifying the active ingredients, putting them into a test tube, and putting the system back together. So the equivalent for neuroscience, of course, is this type of behavioral reconstitution. The second locked door that optogenetics I thought would allow us to open is the search for anatomical connections between neurons, which, if done with electrodes, is a very painstaking process. But if one could replace these electrodes with surging light beams that can be rastered quickly across the tissue, then, of course, the throughput and also the molecular specificity of the approach would increase dramatically. And finally, if one had an idea about how something worked, a mechanistic hypothesis, then of course the way to find out whether that hypothesis is right or wrong is through targeted interference with the system. So for the rest of the lecture today, I've chosen to illustrate these advantages that optogenetics has brought to our work through one topic from our recent work that I think combines all 3 of them, and that is the neuronal control of sleep. Sleep is one of the great biological mysteries. Each night we disconnect ourselves from the world for 7 or 8 hours, a state that leaves us vulnerable and unable to do what we need to do in order to propagate the species. Yet, despite these risks and costs, we still don't know what sleep is good for. And one way to find out what it might be good for, what the vital benefit of sleep might be, is to investigate the control mechanisms that normally regulate sleep and waking. And there's 2 of them in the brain that are symbolized here by these 2 different types of oscillation. The sine oscillation symbolizes the circadian clock, which oscillates in synchrony with predictable external changes in the environment that are caused by the Earth's rotation. As such, it's a purely adaptive mechanism that makes sure we do our vital sleeping when it hurts us least. But I think it cannot speak to the deeper mystery of why we need to sleep in the first place. The answer to that deeper mystery will in all likelihood come from understanding the second control, or the sleep homeostat, which is illustrated here by this sawtooth oscillation that's superimposed on the clock. While we are awake, something, and we don't know what that something is happens in our brains or in our bodies, and that something accumulates up to a certain threshold. And when that threshold is reached, we go to sleep. The system is reset during sleep, and when we wake up, the cycle begins anew. Now, we know a lot about the circadian clock, and this paper is really the Rosetta Stone that broke the problem open, the discovery by Seymour Benzer and his graduate student Ron Konopka 45 years ago of fruit flies whose circadian clocks ran abnormally fast or slow. From this initial discovery flowed a pretty complete molecular, cellular, and circuit understanding of circadian timekeeping, and the three key individuals in that field were actually recognized with the Massry Prize in the year 2012. Now, this slide, in contrast, summarizes pretty much what we know about the sleep homeostat. And my goal for the rest of the talk will be to try and draw some outlines on this blank canvas. The story begins with the discovery by a former postdoc in the lab when he was a graduate student in Paul Shaw's lab at Washington University in St. Louis. The name of the postdoc is Jeff Donnelly. He's sitting right over there because he recently joined the neurobiology department at UCLA. So what Jeff discovered were neurons that are shown here in three expression patterns whose artificial activation would promote sleep. And I would like to draw your attention in particular to the expression pattern on the lower right, which is the cleanest pattern. It captures only these 24 or so cells, about 12 in each hemisphere of the brain. And you can see the axonal projections of these neurons into a structure called the dorsal fan-shaped body. The way we do the experiments to analyze the function of these cells and discover the causal roles they play in the role of sleep and waking is to head fix a fruit fly. Our work is also done in flies, just like Benzer's, and monitor its movements by having it walk on a spherical treadmill. Since there are no known cases of somnambulism in flies, we know that when the fly is walking, it must be awake. And at the same time, we record the activity of one or more of these sleep-promoting cells through a patch pipette, and we express our optogenetic actuators, our light-gated ion channels, in the entire population of these 24 cells. And when we perform an experiment like this, you can see that if the fly, in this case, starts out walking along happily while the sleep-promoting cells are silent. As soon as we switch on the lights, the sleep-promoting cells begin to spike, and all movement quickly ceases, consistent with the fly falling asleep. When we take the drive away, the cells fall silent, and movement quickly returns, suggesting that the fly wakes up. If we switch the cells back on, we can put the fly back to rest. So we have isolated, or Jeff has isolated, a switch in the brain of the fly that allows us to toggle the animal between sleep and waking. And through a series of experiments that I don't have the time to review, we then finally discovered that these neurons actually have nothing to do with the output of the circadian clock. They represent the specific output arm of the sleep homeostat in the brain of the fly. Recording from these cells, and here's one such cell shown filled with a fluorescent dye through a patch pipette, we found that they are anatomically an extremely homogeneous population. Each cell that we patch looks exactly like this. Each cell innervates the entire stratum of the dorsal fan-shaped body. So there's no real anatomical distinction between one of these neurons from another. But physiologically, they are probably the most heterogeneous population that we've ever characterized. Here's an example of recordings from two of these neurons in two different flies. On the left, the cell looks pretty much like a well-behaved neuron. You see that the cell responds to the injection of steps of depolarizing current with action potentials. The frequency of these action potentials increases in a graded fashion with the amount of current that we inject. Everything behaves as it should. And on the right, this cell doesn't look like a neuron at all. You can see we can step depolarize this neuron to positive membrane potentials and still not squeeze a single action potential out of it. If you look at the passive membrane properties, these are all standard-sized current steps, you can tell that the input resistance that this cell puts against the current that's being injected is large, because the same current step causes a large voltage deflection, whereas in this case the voltage deflection is small. It also takes this cell a long time to settle into a new equilibrium membrane potential after a current injection, suggesting that the membrane time constant is long, whereas in this case the membrane time constant is short. So together, the combination of low input resistance and short membrane time constants is virtually diagnostic of a shunt opening, a leak that dissipates the inputs that the cell receives that try to drive it. So when we observed these phenotypic differences between the biophysics of these neurons and convinced ourselves that they were real, then of course we thought, well, maybe this is the mechanism of homeostatic sleep control. Maybe these neurons switch between these two states, a state in which they're electrically active in a fly that's sleeping, and in another state in which they're electrically silent in a fly that's awake. So, of course, in order to substantiate this idea, what one would want to do is actually capture one of these neurons in the same fly, switching between these two states, really observing it going from an electrically active to a silent state and back, correlating the state of the neuron with the behavioral state of the animal, its sleep or wakefulness. Ideally, one would, of course, also want to know the signals that normally operate this excitability switch. And if one had a signal that one could apply to flick the switch in one direction or another, that then, of course, would be the experimental prerequisite for understanding the mechanism that underpins it all. So what might such a crucial signal of regulating the state of these neurons be? Well, a hint had come in the first optogenetic experiments in which the behavior of an animal was remote controlled. These were done by my then graduate student, Susanna Lima at Yale in 2004. And what Susanna had done was she measured the movement trajectories of fruit flies in a circular arena. So each of these black tracks represents the walking path of one fly over a period of 2 minutes, before and after she switched on optogenetically all dopaminergic neurons in the brain. And you can see from the massive increase in locomotion that dopamine has a strongly arousing effect on the behavior of the fly, as it does in mammals. All psychostimulants, from cocaine to amphetamine, exert the arousing effects by elevating synaptic dopamine levels. So if dopamine is an arousal-promoting signal in flies, you would, of course, expect that that arousal-promoting dopamine acts on the sleep-promoting neurons and shuts them off. So this is the idea that we thought we might be able to test using optogenetics. In fact, there is a dopaminergic projection that targets the same brain region that's also inhabited by the sleep-promoting cells that Jeff had identified. In fact, the arborizations of the two classes of neurons shadow each other so closely that the question, of course, immediately arises, are the two of them connected? And we can probe for connections between the two cell types by putting a light-gated channel into the putative presynaptic neuron, in our case the dopaminergic cell, and recording electrophysiologically from the putative postsynaptic partner, the sleep-promoting cell. And what we would expect to see, of course, is inhibition of the postsynaptic partner and ultimately a state switch from the electrically active to the electrically silent state. And here's such an experiment in which we record the behavior of one of these sleep-promoting cells. Now it's the same cell in the same individual. The experiment was done over a period of roughly 1 hour. You can see the cell starts out in the electrically active on state in a fly that's sleeping. Then we apply dopamine by optogenetic stimulation of these neurons, and you can see that the cell switches to the electrically silent state. The ability to produce action potentials has disappeared, and you also see the passive membrane changes. You see the drop in input resistance. You see the shortening of the membrane time constant. Now, this is not damage that we have afflicted onto our neuron through our experimental manipulations, because if we wait long enough, at some point spontaneously the cell flips back into the electrically active state, which would correspond to the fly going back to sleep. So this temporary suspension of all electrical activity is not a dead end that's been brought on by our experimental condition, but it's part of the normal duty cycle of this particular cell. the action of dopamine on these neurons is direct, because if we take the dopamine receptor that mediates these effects, one particular receptor out of 4 in the fly's genome, if we take that receptor away selectively from these 24 sleep-promoting cells, you can see that we can block the switch completely. So both the change in input resistance and in membrane time constant is completely abrogated. You will also notice from this kinetic plot that the change in these parameters happens on a fast time scale, on roughly on the order of about 1 minute. This time scale is, of course, way too fast to be accounted for by the de novo synthesis of ion channels. Instead, it must be reflecting a modulation of the channels existing, of the cell's existing channel inventory. And I get to the mechanism of modulation in just a second. Now, if we switch the physiological state of these cells in a fly, can we also switch its behavioral state? The answer in a nutshell is yes. As you see here, these are movement rasters of 6 flies that all start out sleeping. So you see they don't move, with the exception of the occasional twitch that's still consistent with sleep. And at time 0, we start to apply dopamine. And we stop the dopamine delivery, indicated by the transition from red to black, when the input resistance has dropped to roughly 2/3 of its initial value. What you can see is that the switch has a memory. It does not spontaneously flip back, but rather once you've actuated it, it stays, in this case, in the off position for an extended period of time, even if you stop the dopamine delivery. And the fly stays awake and starts and keeps moving for equally extended periods of time. With one exception, the case shown here with an arrowhead, where all movement stops at this particular time point, and we know from the electrophysiological recording that was performed spontaneously that in this case, the sleep-promoting cell flipped back into the electrically active state. In other words, the animal just went back to sleep. Now, what might be the mechanism that underpins this form of biophysical switching behavior of the neurons? What we have is a neuromodulator that acts on a particular type of receptor that then couples through a signal transduction cascade to channels that are either modulated in their gating or conducting properties, or that are modulated by changes in membrane presence. There's precedence for many receptors or transporters that get modulated by varying the number of copies in the plasma membrane using endo and re-exocytosis of channels into vesicles. Now, some experimental data that again will skip in the interest of time had pointed us in the direction that potassium conductances were the critical players in this process. So our further analysis focused on the analysis of potassium currents, which we did in the classical John Connor and Chuck Stevens way by doing voltage clamp experiments, starting from different holding potentials to separate 2 types of potassium currents, namely voltage-dependent A-type currents and voltage-independent non-A-type currents. You can see that our sleep-promoting cells express both types of these currents. The non-A-type currents you see at the tail end of these voltage steps, and the voltage-dependent A-type currents as this fast, rapidly inactivating transient at the beginning of a current step. If you just replot this current-voltage relationship, you can see the same thing, so that the sleep-promoting cells have A-type and non-A-type currents with characteristic biophysical properties. If we take now a cell in voltage clamp through the same optogenetically induced dopaminergic switch in its excitable state, what we've discovered to our surprise was that not one current changed, but both changed, and they changed in opposite directions. So the non-A-type currents increased about 4-fold when measured between resting membrane potential and spike threshold, whereas the voltage-gated A-type current dropped by about 1/3. Again, the changes were completely reversible, indicating that the switch actually can be flipped in both directions. So when I initially said that we were hunting for a modulated conductance, that of course was incorrect, but because we have to find at least 2 such conductances, one voltage-gated A-type current and a voltage-independent non-A-type currents. Now, the logic that we applied in our search for these conductances is simple. If you have a system that can switch between these two states, an electrically active on state and an inactive off state, and these two states are dominated by two different potassium currents, a voltage-dependent A-type current in the on state and the voltage-independent non-A-type current in the off state, then by taking away the current that dominates the respective state, you should be able to push the switch in one direction or the other. So, for example, if you take away the A-type current that's necessary for the on state, you should push the sleep-promoting neurons in the off state. And since they cannot become electrically active, the behavioral consequence of this intervention should be a reduction in sleep, since these neurons are required for sleep, and vice versa. So we went through the complete set of voltage-gated potassium channels encoded in the fly's genome, and knocked them down selectively in these sleep-promoting neurons using RNA-mediated interference. And as you can see, we found two, the classical A-type current shaker and a close relative called SHAB, whose loss from these neurons caused a dramatic loss in sleep, as predicted. Now, for the other type of current, if you look at this current-voltage relationship, it has the almost cartoonish shape of a passive leak conductance. There's very little rectification. It's an almost perfectly linear relationship between membrane potential and current that flows. The reversal potential sits at -80, which is, of course, consistent with potassium being the permeant ion. It looks like a cartoonish leak conductance. And these leak channels are widely thought to be formed by a family of potassium channels called 2-pore domain or K2P channels, of which fruit flies possess 11 in their genomes. And again, we took them out one by one and found one of these channels that caused the predicted increase in sleep, which is the opposite effect of taking away the voltage-gated shaker channel. That channel had not been characterized in any way. It just had emerged from annotation of the genome sequence of the fly. So in following the tradition where gene products in fruit flies are named after the mutant phenotypes, and noting that some of these flies literally doze away the vast majority of their existence, 23.5 hours of sleep a day, we decided to name this gene product and the ion channel Sandman. Now, what you would, of course, predict what Sandman does is that it interferes with the physiology of these sleep-promoting neurons in a way that prevents them from switching off. And that could then account for the hypersomnia that these animals experience. And physiologically, that's exactly what we see. If we take Sandman out of these sleep-promoting neurons and take them through the optogenetic application of dopamine that switches a control cell from the electrically active into the silent state, you can see that in cells that lack the Sandman channel, the switch is broken. It's broken to the same extent as in cells that lack the dopamine receptor that transduces the arousing effects of dopamine. Now, if we quantify the amount of currents that flow after our RNAi-mediated knockdown in these cells in voltage clamp, you can see that as far as the A-type currents are concerned, there's no real surprise. If we knock down shaker in these neurons, the voltage-gated A-type currents in both states are reduced by about 1/2, which is what you would expect based on shaker not being the only voltage-gated potassium channel being expressed in these cells, and of course also RNAi not being 100% effective. You can see that the switch, or the attenuation of the A-type current as a function of the on-off switch is still intact, so the remaining current is properly modulated, and interference with Sandman has no effect on voltage-gated A-type currents, consistent with the idea that this encodes a different type of potassium channel. However, if we look at the non-A-type currents, there's a little surprise in store. And that surprise is that if you take Sandman away from these neurons, you don't see any effect in the electrically active on state. But you see that the increase in Sandman current that occurs after switching the cells off is completely blocked. So what this means, of course, in the scheme of our regular, in terms of mechanism, is that the Sandman channel is either completely inactive in the electrically active state, or it's not present in the plasma membrane because it's completely internalized in external vesicles and gets inserted into the membrane only after dopamine switches the cells off. So how could we distinguish between these two mechanistic possibilities? Well, we decided to include in the patch pipette a botulinum neurotoxin, which cleaves SNEA proteins that are required for vesicle exocytosis. And what you can see is that this experimental manipulation fully duplicates the effect of taking Sandman out of these cells. And of course, the conclusion of these experiments is that it's this scenario on the left here that describes reality. So Sandman actually actively translocates between intracellular vesicles and the plasma membrane in response to dopamine. Now, the loss of Shaker, of course, also must cause the sleep disruption, must have a physical basis for the sleep disruption that it causes. And that physical basis is the opposite of what Sandman does. If you take Shaker away from these neurons, the cells switch off perfectly fine, so the flies have no difficulty waking up. But what's defective in these neurons, as you can see from these anecdotal examples here and from the population data shown here, is that these cells cannot maintain high-frequency spiking. And that, of course, means that they cannot produce the electrical output that's required to put these flies asleep for extended periods of time. So what I've described to you then is, I think, a pleasing symmetry or anti-symmetry between two potassium channels that are modulated antagonistically, and their reciprocal effects on the biophysics of selected groups of neurons and a complex organismal behavior such as sleep. So it's an almost seamless voyage from organismal physiology to ion channel biophysics and back. By way of summary, the device that I've introduced to you is similar to one that you're all familiar with because you have it on the walls of your living rooms, except that this device does not measure temperature and turn on the heat when it's too cold. It measures sleep need and puts you to sleep when your sleep need exceeds a certain set point. The billion-dollar question in all of this, of course, is what is the equivalent of temperature in the system that I've just described to you. Before I conclude, I would like to take you back for just 1 or 2 minutes into the stone age of optogenetics. When we started to work on the first photoreceptors for use in neuronal control, I became aware that I was not the only scientist who had seen the need for a technology like this. And I became aware of this because of a citation I learned to an earlier of my papers. The other scientist was Francis Crick, who, in a paper published in the millennial issue of the Philosophical Transactions of the Royal Society and called The Impact of Molecular Biology on Neuroscience, wrote, "The next requirement for understanding the brain is to be able to turn the firing of one or more types of neuron on or off in a rapid manner in the behaving animal. The ideal signal would be light. This seems rather far-fetched, but it is conceivable that molecular biologists could engineer a particular cell type to be sensitive to light in this way." So when we had the first experiments that turned this far-fetched possibility into a reality, and these are these experiments, what you see here is a hippocampal neuron from rat grown in culture. The neuron expresses an opsin protein along with GFP so that we can patch it. And when we record the membrane potential of this cell in the dark, you see it hovers around resting value. And as soon as you switch on the lights, there's a depolarizing shift, and the cell responds with a volume of action potentials. So when we had these experiments, I decided to send Crick a preprint of our paper. Now, if you read this wonderful book called The Eighth Day of Creation, which recounts the early days of molecular biology, you know that Crick was a prolific letter writer. He steered the development of the field through a vast correspondence, whose stylistic hallmarks were always encouragement and constructive criticism. And that's exactly what I got. Crick wrote back to me to say that he read the paper I had sent him with great interest and was excited to see that the system already works, at least to some extent. However, he realized, as I did, that it still needed improvement and that this would take further work. Unfortunately, Crick did not live to hear how our experiments progressed, but the improvements certainly have come to a very significant extent through the work of my co-laureates, Peter and Carl. So I think this is the appropriate moment for me to step aside and turn over the stage to them. But I think it's fair to say that as a result of our collective efforts, the way we neuroscientists approach our problem has fundamentally changed. I'd also like to acknowledge the people in my lab who have been responsible for this work. The current crew is aligned to the left, and some of the notable alumni have been displaced by one tab stop to the right. Boris Semmelmann was the postdoc who made the first optogenetic actuators. Susanna Lima was the graduate student who first used them to control the behavior of an animal. And all the work about sleep control that I talked about today was done, of course, by Jeff and another postdoc, Diogo Pimentel. Thank you.